Question 5. The diagram shows some features of a plasmid which have been cut open by a restriction endonuclease to allow a gene from a donor chromosome to be inserted. Okay, the DNA recognition sites for 3 restriction endonucleases, EcoR1, BAMH1 and HIN3 are shown in the table. The arrows indicate where each restriction endonuclease cuts the DNA sequence. Okay, that's fair enough. You've been given information here, and then on the plasmid they've already done the cut, and then we've got the ampicillin resistance gene and another one which have not been named, and as soon as you see that you're like, they're going to ask me what that is. Okay, a restriction endonuclease was used to remove a gene from a donor chromosome. Use information from the diagram and the table, so you're being told, and that's in bold, you're going to have to use both, to identify the restriction endonuclease which would be used to allow the gene to be inserted into the plasmid. Give a reason. Okay, so if you were totally panicking in the exam, you've got a 1 in 3 chance of getting that first mark, even if you really hadn't got a clue. But obviously you're never going to get the second one unless you can see what's going on. So let's have a look at what we've got here. You've already had this cut, and what you've got on the cut here, these are your sticky ends. You've got an AATT and a TTAA. I need something that's going to give me the complementary sticky ends for that, otherwise I'm not going to be able to stick it. Okay, so basically here's what I need. Here's my restriction cut that will give me exactly the same one, because this will cut down through here and along and down, and that means I will have complementary sticky ends that will work. Okay, the other ones won't, because I'll get a GATC and a CTAG and a AGCT and TCGA. Okay, so restriction endonuclease I'm going to use is EcoR1, and reason, I get complementary sticky ends. Name the enzyme that would be used to seal the gene into the plasmid. Well, that's a bit of a gift. That's ligase. When I say gift, you obviously need to know the answer. Okay, a culture of bacterial cells, 20% of which had taken up this modified plasmid, were grown in a nutrient agar plate. The plate was incubated and 250 colonies of this bacteria grew. Predict the number of colonies which would have been expected to grow if the nutrient agar plate had contained the antibiotic ampicillin. And you got a space for calculation, which I think is, I don't know how much you would need to use this. You could get away without even a calculator on this one. Okay, we have 250 grown. 20% of them have taken up the ampicillin resistance gene with the plasmid, so 250 just times that by 20%, obviously 0.2 in your calculator, so you would get 50. Or you could have said, I'll divide by 10 to get 10% and times by 2 if I was doing it in my head. Okay. Name region X shown in the diagram which ensured that the modified plasmid would be passed on to daughter cells. So the whole problem with plasmids being added in is if they don't then replicate, so you've got multiple copies, when the bacterial cell divides, it won't go into the daughter cells. So what you need is an origin of replication. And that is what region X is, or given as RI. That's it.